Okay, so it gives me great pleasure to introduce our second speaker, again, a good friend for a lot of us, and one of the leaders of the trial that essentially established the first standard of care and thus transformed how we treat biliary cancer, established a good proof of principle, I think, that we can actually take a rare disease and apply a good, well-designed phase 3 study and be able to transform care. So I think he had been the catalyst for everything we do today, with a lot of hope about how we are able to transform piece by piece this cancer into a more curable disease. So Dr. Bridgewater comes from the UK. You'll notice this by his accent. Actually, he a little story on the side. I know you still have to catch a plane, but I have to say that. So he came, visited us, and my kids just adored him. You know, he was just lovely. They loved his accent. Not that I don't have one, but it's not as lovely. And they essentially were promised that every year he will show up on his way to this conference. He didn't show up this year. I told my kids I'm going to Salt Lake City for the conference. So first question they asked, this is a year after. My oldest is 7, my youngest is 4, so you know, these are young kids. They said, where's Dr. Bridgewater? With a really bad fake English accent. So I promised them next year, John, that you'll pass by, and I hope you do. So without further ado, Dr. Bridgewater will be updating us about integrating genomic signals into clinical trial designs for biliary cancers. John.
Thank you, Tony. How on earth do you follow that? Well, here we are. So first of all, actually, thank you very much, Jennifer and Stacey, for inviting me. Always a great pleasure to be here. This is a very special conference because of all the people sitting over here. And it puts a different complexion on things. You learn different things when you come here. You learn how to make it really count. Perhaps that's not the best way of putting it. Day 1.5, you'll have heard a lot of stuff, and I'll try not to repeat any of it. There's a small risk of doing so. And we'll start with the simple stuff. I know some people find it difficult to, some of the science can get a bit heavy. Cancer is a genetic disease. When the genes go wrong, the cell goes into overdrive, and you get a huge number of messages going to the cell nucleus and a proliferation of the cells. And what we do is find drugs that stop that. And that's the whole paradigm behind finding a target for the cancers we treat. This is really easy when you have a one-gene cancer, and these are examples of them. There's cancers called GIST and acute myeloid leukemia, in fact, where there's a single abnormality, and you can have a single drug that just takes out that abnormality. And this is the famous drug Gleevec, potentially the first of these targeted therapies, which worked really well. And it did so because the situation was simple. In reality, it's not really like that, and you know, artificial intelligence will have to work its way around this. This is more like what happens. This is the famous Tokyo underground map, but actually probably resembles the way in which our cells interact much more than we'd like to think. So several steps beyond the one gene, one target, one drug dogma. That doesn't really work in this situation. There are further complications. There are probably several different cancers in every tumor mass. This is Charlie Swanton's work looking at originally kidney cancer, but then lung cancer. And you take lots of different bits of this cancer, and within the cancer itself, there are different cancers develop, and he's done incredible work showing how they actually link to each other. One comes from the other, one comes from the third, actually skips one cancer. So how do you is it rational to treat 4 cancers in the same way? Of course it isn't. The other big problem we have here is the long tail of cancer genes. So if you have a targeted gene, so in fact this is B-Raf, so about 1 in 200 of these cancers, and this cancer happens to be AML, it's acute myeloid leukemia. 1 in 200 acute myeloid leukemias are caused by B-Raf. There's no question about it. It's the troublemaker here. Now, how do you design a study? How do you design a strategy for actually showing that B-Raf drugs work in 1 in 200 patients with this disease? So that's yet another challenge for the way in which we try and target molecular abnormalities. Well, starting early with collaboration rather than leaving it to the last few slides. This is the most recent and biggest collaboration to actually decipher, to work out, to list all those genetic abnormalities in cholangiocarcinoma. And this was the International Cancer Gene Consortium, based in Singapore, in fact. But with lots of international partners. They can be criticized in many ways. Only some bits of analysis were done for some of the cancers. They were mostly post-operative cancers, so hardly any cancers from patients with advanced disease, which is sadly what we deal with most of the time. A lot of bias towards the intrahepatic cancers for a number of reasons. Nevertheless, you come out with these chunks, the divisions within cholangiocarcinoma, and really you've seen a lot of these names over the past 2 days, the B2s, the FGFRs, and others, and you can come out with these divisions. And indeed, that was the ICGC and other work. So this is Nabil Debardesi's work, and it says really very much the same thing. And so you have this picture, which again you've seen several times already, where cholangiocarcinomas carve up nicely into several anatomical types that also seem to have quite a specific biological types as well. And the sort of swirly thing on the left just shows which one of these abnormalities coincide with the others. And the P53, so the big blue bit connecting with the big blue bit. Now, I haven't got a pointer, so I'll have to describe. Tells us that the P53s co-occur, so they occur most commonly with the KRAS genes. Everything else doesn't really occur with each other, and that gives us a clue as to the nature of this cancer. If you occur by yourself, if you're the only troublemaker in the cancer, then you're quite likely, then it is going to be easier to target you than if you were mixed in with a number of other abnormalities. And I'll just give you a few examples of that. Most of you will have, some of you will have had this test, most of you will have seen one of these tests. And I'll take you through how you try and figure this out. The sort of Greek chorus on these occasions, the mantra is, oh, show it to the genomics board. Most of my genomics board won't have seen much of this and are completely useless when it comes to telling me what to do about some of my patients. So it is a question of very much working it out as we go along, and I'll show you how that really is the case towards the end of my talk. So this is easy. This patient, if you look at the sort of bottom right box, that's the nitty-gritty. That tells you what the matter is, and that tells you that this patient has an FGFR fusion. The PI3 kinase isn't really treatable these days, and the check is very experimental, and I have no idea what the other thing is. So really this was very straightforward. The patient went on to an FGFR study. What happens here? Well, it tells you very nicely that there's an IDH1 study. There's the PI3K thing again, and that's not very treatable. There is a B-Raf mutation, D3P14, so not the 600Es about which we heard earlier, a sort of slight variant on that. What on earth to do about that? I'm not entirely sure. And in this patient, I was, what do I try and treat? Do I treat the B-Raf? Do I treat the IDH1? Actually, we had a study for IDH1, which we all know about, and the patient went into the IDH1 study. Was that the right thing to do? I'm not so sure. And this is even more complicated. So this is the page 1, and you get all this stuff come out, and then you have to look at the also-rans. So about 5 or 6 pages down, you get the also-rans, unknown significance, and there was a little box here saying FGFR rearrangement, and it took about 3 or 4 weeks of questioning Foundation Medicine to determine that actually this was probably a fusion protein for which they did not have the correct pairings and should have really moved up onto the front page, but didn't. So these are not straightforward things to work your way through. Having said that, we have a nice list of targets, and I'll try and explain how we're trying to combine that into one study. This is the French Moscato data which you saw yesterday. So Moscato was a big sort of, in fact, match-batch type study where over 1,000 patients were treated. They're treated on this ICSI program, which is the French expanded use program. So they get use of all sorts of drugs on this program. And of 43 patients, 43 bile duct patients, 23 had targets. That's pretty much what we think is. We think it's around that sort of level. Of whom 18 were treated, and there were 6 responders. So the blue guys on the far left-hand side, 6 responders. That's pretty good. 6 out of 18. Now remember that when you look at the bigger population of the Moscato study. So 1,035 patients, of whom 843 tests were performed. 193 had druggable targets, of whom 22 responded. 6 of those 22 were the bile duct patients. So you can see that cholangiocarcinoma in this way is special. It's special. It's got more targets. It's potentially more treatable. This is very exciting for people like me. What were those targets? So if you just read the small print on the far right underneath those blue lines, this is what they were treated with. So actually, the FGFRs, yes, we've come across those. The ERB2s, yes, there we go. And then there was an EGFR amplification as well. So really, it's all of a sudden, it doesn't seem terribly complicated. This is what we've got to go for. And you've seen all this stuff about IDH1s. I won't talk about that. These are the FGFRs. Won't talk about that. Heard a talk about this earlier, so I won't talk about that. Just to mention the immunotherapies have been pretty disappointing. What do? We do this. So this is what I was asked here to talk about, really. This is what Jennifer wanted. So I'll spend a little bit of time on this because it is horribly complicated. If we start at the top, we have 800 patients. And they all have currently Foundation 1 testing, and then they all have 3 months of chemotherapy. Currently, that's cisplatin gemcitabine. We may have to change that to FOLFIRINOX when the French study reports in about 18 months' time, if it happens to be positive. A proportion of these will have a successful profile, and a proportion of those will have a identifiable target. So the targetable ones is sub-study number 1. So if you, this is where I really need a pointer, but the first green box on the bottom line, called sub-study number 1, is the heart of this study, is where we compare those patients with recognized alterations. We've said 40%, Moscato said 63. We said 40%, and these patients will be randomized between standard of care, so that'll be more cisplatin gemcitabine or whatever chemotherapy you have, and then you'll and then on progression or the targeted agent, and on progression they'll either cross over into another target, they may have more than one target, or go back to the chemotherapy. The second box, sub-study 2, is those with sort of questionable alterations. So I'll show this much more on the second slide. No, why don't I show the second slide now? This is the second slide of greens and reds. The greens are probably the better bets here, okay? So these are the ones where we think we're on pretty firm ground, and we're going to treat the patients with this. This is the heart of the study. If this bit doesn't work, we're toast. You know, this has got to work. The red bit is the much more questionable bit. The red bit is, well, this looks like a good target. Do we have a drug? Maybe, maybe not. And we can consider this as the red bit as really multiple experimental phase 2 studies, you know, just single-arm phase 2 studies. The red bit is not going to be randomized. So that's sub-study 1. So the green bit is sub-study 1. The red bit is sub-study 2. Sub-study 3 are patients who progress on cisplatin and gemcitabine and happen to have a target, and they will get the red or green drugs as appropriate. So we had to put these in because we ran this past Helen Mormont, our extremely wonderful user rep, who said, you can't drop these people. They've gone into your study and in good faith, and they've progressed on treatment, and you're going to say, bye-bye, you can't do that. So that's the arm. Sub-study 3 is based completely on that. Sub-study 4, we're almost there. You've done incredibly well. Sub-study 4 is where you don't have a target or the profile fails. So I'll come to the profile failing bit in a second, where you don't have a target. And here people get immunotherapy or standard of care or immunotherapy and radiotherapy. We know that immunotherapy doesn't work very well. So and the more data that comes out about that, the more reluctant I am to do that. So that may well have to change. So this is it. This is the magnum opus. This will hopefully determine, the main aim of this is to determine whether targeted therapies are worthwhile. The French data so far would suggest that it's not, but I think in this cancer, in cholangiocarcinoma, our special cancer, I think it's going to work. So that's that. And those are the reds and greens. And now we'll move, what am I worried about? This is what I'm worried about. So these are the biopsies that you would get. And actually, in all of that, and this is where I really, really needed a pointer, but the amount of cancer in there is actually relatively small. There are 2 small pink bits in the top blue patch and a couple of small pink bits in the bottom blue patch. And this biopsy would almost certainly have been rejected by Foundation Medicine. And so that's what I'm worried about. What is going to happen to these people? I don't know. The blood tests are nowhere near as good currently. I think that's paraphrasing Mitesh. So that's the one thing I'm worried about. I'm worried about a host of other things. This is what I'm also worried about, the competition. There's an awful lot of competition out there, soaking up the patients, mostly looking at FGFR, but probably other things as well. And at that point, I'll stop telling you what I'm worried about and move on to, oh yes, there are others, and move on to this, which is the sort of story just to finish off, really. So this was a 70-year-old man who had a B-Raf 469A. So again, not one of the 600Es, the famous 600Es you heard about earlier. And he had an intrahepatic cholangiocarcinoma, and he had an operation. I gave him some capecitabine because that's the sort of thing we do, and he progressed. He just developed more cancer, and it was terrible. And then I gave him some cisplatin and gemcitabine because, again, that's what we showed works, and it didn't work. It was terrible. And at this point, I was feeling fairly rough about this, and I'd read up that a combination of molecular targets, that used in the colorectal beacon trial, worked well against certain B-Raf mutations, not actually this B-Raf mutation. After he'd started on the therapy, I asked Mitesh, what shall I give him? And Mitesh said, oh, you need a SARC inhibitor, stupid. I paraphrase perhaps, but that's what he meant. But it was too late. He'd already received this stuff, and thank goodness it worked. So this is the first thing that's worked for this guy. And I'm extremely grateful that he has. His life has been restored. So this is why we do it. This is why we do it. This is, this combination molecular therapy really, really works. So, summary. I think we have a special cancer. It's a paradigm for targeted therapy. Getting the study right is absolutely key. We've got the right targets. We have some very good drugs. We may not have perfect drugs. We have very good drugs. And then making sure the study is designed sufficiently well to show that it works, that's going to be the tricky bit. And the providers need to adapt. I mean, this is an uncommon cancer. Inevitably, the number is going to be small. We will need to persuade them that we're not going to have big numbers to show big results the whole time as we've had before. How can we collaborate? We need to just educate. We need to treat where. So what is a profile? How do I get one? I've heard that so many times the last couple of days. We need to treat. Where do I find the treatment? What treatment should I have? What's the advantages in a trial? I don't need to tell the people in this room, but there are a lot of people out there who don't know this. And assemble the data. There are huge amounts of data, and we must assemble it properly. So thanks very much to everybody. Cancer Research UK, people who give me most money, Helen and her works, various universities and institutions, and of course, Stacey. Thanks very much indeed.
Okay. All right. So because Dr. Bridgewater will have to head to the airport soon, we're actually going to take questions just specific to his talk, if there are any. Otherwise, we'll go back at the end without John. This is a fantastic, of course. In between this and batch, excellent examples of, industry along with NCI and your equivalent in the UK and academic collaborations that will advance the field forward. So thank you. Jan.
Thank you, both of you, excellent talks. So John, before you go, so you have drugs from a number of companies. So how are you working out? Just tell us logistically how this has worked, that you've got them to cooperate and provide the drugs. How many contracts and...
A lot of persuasion. We have persuaded them that this is the best deal in town, and that this study, unlike, I mean, our FGFR colleagues are wonderful, but they have a study for a tiny proportion of patients. This study provides something for everybody. And in that, and that's the main message. That's what we've gone back to them again and again. So far, they've been extremely positive.
My question is, have you considered, or would you consider adding another arm for some of these drugs, targeted drugs, for people who've already been on a prior one? So, for example, you have the TAS-120 to target FGFR2. What about looking at patients? You're worried about, people, other companies soaking up that population, but there's a population out here of people who've been on the infigratinib or pemigatinib. So, could you speak to that?
So this study is a sort of maintenance study. You get the treatment after a bit of chemotherapy. You can then go back to the chemotherapy if you want. So probably people who've had other targeted therapies before that probably wouldn't qualify. That's a very good question, though. I mean, what are we going to do about these people who progress on one thing and have to go on to the other? And there's a whole other series of talks about how we're going to sequentially biopsy people here and work out what next to give them. If we can't do that in this cancer, we won't be able to do it in anything.
One more question. Last one.
I just have one more question. Have you considered adding an arm for patients who have some of the comorbidities that exclude them from other trials?
Some which?
So comorbidities.
Oh,
So
I
they
see.
get excluded because they have ulcerative colitis or some other thing. Have you considered adding an arm for some of those, to make options for some of those patients?
So we did do this. We considered putting in essentially an expanded use program as an addition to it, and in the end, this proved a bit difficult. We may come back to it though. Now that you've mentioned it, thank you very much.
Okay. Thank you, John. See you, my friend. So this is the other one. This was wonderful. Have a safe flight back home. We'll see you soon, hopefully. All right, so again, Shishir.